sleep patterns emerge as a predictive data source for disease risk in new analysis. This is Planetterrian Daily, episode 152. A feature in Nature Medicine positions sleep as a rich data source rather than only a symptom when disrupted. The analysis highlights how sleep metrics can forecast multiple diseases and function as a therapeutic target. Researchers note that viewing sleep this way opens avenues for earlier intervention across conditions. The piece draws on recent clinical observations linking sleep architecture to disease trajectories. Next steps include integrating sleep monitoring into routine risk assessment protocols. Now, shifting to a very different area of research. A new review in Nature Medicine outlines paths to prevent type 2 diabetes and achieve remission once glucose intolerance appears. The work draws on mechanistic heterogeneity observed in large cohorts and emphasizes restoring normal glucose regulation before full disease onset. Authors from institutions including UKT Tübingen detail how timing of interventions matters across different life stages. The analysis integrates data on prediabetes progression and remission rates. Follow-up studies will test targeted lifestyle and metabolic approaches in defined subgroups. Metabolic health and reproductive longevity both hinge on timing. Research published in Nature Aging reframes the ovarian reserve as an actively regulated system instead of a passive reservoir. The study identifies principles that govern female reproductive longevity and opens new research directions. Mouse and human data support the shift in understanding. Authors state the work opens new avenues for understanding the principles governing female reproductive longevity. Future experiments will probe regulatory signals that could extend reproductive span. The same journal paired this finding with direct evidence that exercise and a circulating protein can slow ovarian aging. The Nature Aging study on ovarian reserve reframes reproductive longevity as an actively controlled process rather than simple depletion. Exercise and adiponectin pathways emerge as modifiable levers in both human cohorts and mouse models. This work moves the field closer to interventions that could preserve function rather than only documenting decline. One open question remains how these regulatory signals interact with broader aging programs already tracked in other tissues. Two large cohorts totaling approximately 165,000 participants, combined with mouse studies, show that exercise slows ovary aging. The work also identifies adiponectin as a potential therapeutic target. Findings demonstrate measurable preservation of ovarian function under these interventions. The data link physical activity directly to reproductive tissue maintenance. Researchers plan to test adiponectin modulators in additional models. Now, here's something most people get wrong about the lungs. Most people assume the lungs stay sterile except during infection, yet new evidence shows resident bacterial communities occupy healthy lung tissue. The work challenges the century-old view that lungs remain sterile in the absence of disease. Microbial communities appear shaped by local immune and environmental conditions. The findings expand the map of human microbiomes beyond the gut and oral cavity. Right now, and as you breathe, these microbes interact with local immune cells and airway surfaces in ways that differ from the gut microbiome. The total bacterial load in the lungs is far smaller than in the intestine, yet the species present appear tuned to the oxygen-rich, constantly moving environment. One memorable detail is that these communities can be detected even in people with no respiratory symptoms, overturning earlier culture-based conclusions that reported zero growth. The presence of these organisms may influence how the lung responds to inhaled particles or viruses. A practical takeaway is that future respiratory therapies could deliberately preserve or adjust these resident populations rather than treating all lung bacteria as targets for elimination. From tissue-level disease mapping, we move to a decades-long search that finally yielded a cloned resistance gene in wheat. Proteomic profiling of individual murine aortas identified more than 4,000 proteins per small tissue sample. Proteins including VCAM1, APOB, LPL, and LGALS3 increase progressively from healthy regions toward advanced plaques. Enrichment analysis pointed to extracellular matrix, immune hemostasis, and lipoprotein pathways as drivers. The approach used ApoE-deficient mice on a Western diet for 16 weeks. The method now enables finer mapping of site-specific disease mechanisms. Researchers at the University of Maryland cloned a gene that confers resistance to Hessian fly larvae feeding. The gene product interacts directly with a protein produced by the insect. The finding removes a long-standing barrier to engineering pest resistance in wheat. Experiments confirm the molecular interaction in controlled assays. Breeding programs can now incorporate the cloned sequence into commercial varieties. A technique now allows detailed gene expression and cell type analysis from archived tissue samples. The approach overcomes previous limits on working with older or fixed material. It enables comparisons of cell states across age groups and disease conditions. The method supports studies that were previously restricted by sample quality. Validation work continues on additional tissue banks. Decades of observations across European mountain summits show vegetation moving toward thermophilous species. The direct correlation between this thermophilization and measured local temperature rise proved surprisingly weak. The international study combined field data from multiple countries. Authors conclude that additional factors beyond temperature drive the observed community changes. Continued monitoring will clarify the relative contributions of warming and other pressures. A genetically modified microbe rapidly breaks down abundant minerals to capture atmospheric carbon dioxide. The same process releases critical elements used in electric vehicle batteries. Cornell researchers demonstrated the dual function in laboratory tests. The approach links carbon removal with resource recovery in a single biological step. Scale-up trials are now under consideration. Cells organize reactions through biomolecular condensates that operate across all domains of life. This principle supplements the classical membrane-bound organelle model. Research shows condensates create distinct chemical environments that influence reaction rates and specificity. The framework explains behaviors observed in both normal and stressed cells. Further mapping of condensate rules will refine understanding of cellular decision-making. Live imaging revealed that epithelial cells pulse synchronously as they proliferate and migrate. Malignant cells maintain these pulses for extended durations compared with healthy counterparts. The coordinated behavior supports tissue formation and wound repair. The study links pulse duration to cellular state and invasive potential. Researchers will next examine whether disrupting the pulses alters cancer progression. Blood sample analysis from spaceflight participants clarified mechanisms behind common constipation in microgravity. The University of Copenhagen-NASA collaboration isolated effects that occur without gravitational influence on the gut. Findings point to altered fluid and electrolyte handling in the stomach. The data improve models of gastrointestinal function during long-duration missions. Countermeasure development will incorporate these physiological insights. An implantable cell therapy developed at Rice and Northwestern universities shortens the period needed for circadian adjustment. The construct senses and responds to light cycle mismatches to speed clock resetting. Animal tests showed faster restoration of normal rhythms compared with untreated controls. The approach targets metabolic and sleep disruptions that follow jet lag. Human translation studies are planned. A study of 1 million individuals identified protective variants in the folliculin-interacting protein 1 gene. Carriers showed improved metabolic parameters and markedly reduced disease incidence. Cell and mouse experiments confirmed that loss of FNIP1 function alters nutrient sensing and energy handling. The genetic signal points to a druggable node in metabolic regulation. Therapeutic programs targeting this pathway are now under exploration. Keep an eye on follow-up human trials testing adiponectin modulators for reproductive longevity. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. Quick tip from the network. Try the Do Positive Pod next. Good news in science and tech, plus one concrete action you can take. Every NARA Network show is free at naranetwork.com. Want the written briefing with source links? Each show keeps a summaries page on naranetwork.com, scannable, dated, and free. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.